Beneath your skin, deep within each of your trillions of cells, lies a silent scripter, whispering thread of letters that shapes your being. This code, your DNA, spins gently like a winding staircase of life. Tonight, you step inside its spiraling corridors, and as you listen, you'll come to see. Your body is a living story, written in 4 gentle letters. Close your eyes, let your breath become quiet, like pages turning in an old, familiar book. Your body begins to slow not into stillness, but into listening. You are about to journey inward, not across continents or stars, but through the softest, smallest chambers of yourself, beneath your skin, beneath your bones, inside the warm hush of your cells. Something is writing. Something has always been writing, not in ink, not in words, but in letters, just 4 of them whispered again and again in different combinations, like poems stitched into every living thing. ATC and G. for letters, endlessly rearranged. They are not magic, they are not symbols, they are molecules, tiny shapes that click together like beads on a string. This string is your DNA, and it stretches through you, curling gently in every cell, like a sleeping serpent coiled in light. Your body is built from instructions written in this code, and yet you've never read it not with your eyes. It was written before you were born, and it's being read every second by the quiet machinery inside you. Right now, as you lie still, your cells are reading lines of it, like lullabies, telling your heart to beat, your skin to grow, your thoughts to drift. Let yourself drift too. Imagine zooming inward, past your fingertips, past blood and tissue, down through the translucent walls of a single cell. Inside is a trembling softness, liquid and light. Floating in that ocean is the nucleus, a glowing pearl, and curled inside that pearl is the book of you. You are made of chapters, 23, pairs of chromosomes, each one a bundle of DNA. Stretch them out, and the strands would reach nearly 2 meters. Folded neatly, they sit in every cell, like origami made from code. Your body carries this entire encyclopedia, not once, not twice, but in nearly every cell. It contains around 30 trillion copies of you, whispering in unison. But how does this code speak? It doesn't speak the way we do. It sings in shapes and folds, in loops and triggers. Inside your cells, special molecules called enzymes glide along the DNA, like careful fingers reading Braille. They open up a section, just a few letters long, and make a copy. This copy is like a message, a set of instructions. That message floats to another part of the cell, where tiny factories called ribosomes begin to work. They read the message. They build. What do they build? Everything. Keratin for your hair, collagen for your skin, hemoglobin for your blood, insulin for your sugar, serotonin for your joy. Every feeling, every breath, every blink begins in the code. It is not commanding, not loud. It is simply present, always being read. It is the most intimate kind of storytelling, and yet this story is not new. It has been passed down to you, from your parents, from their parents, and theirs. Your DNA is not just you. It is a quilt of ancestry, a map of migrations, a collection of the victories and sorrows of lives before yours. That freckle on your arm, that particular curl of your hair. These are footnotes from generations past, and sometimes the story includes, at its small random typos, a swapped letter, a skipped phrase. These are called mutations. Most are harmless, some are beautiful. A rare few may bring illness, like a ripple in the harmony. But even those are just lines in the story, waiting to be understood. You are not a machine. You are not software. You are not a perfect draft. You are an unfolding manuscript, and every moment your body keeps reading, shaping, living. Let yourself. Imagine your DNA not as cold code but as a spiral staircase. Step by step, it winds through you, connecting your present to your past. Each step is made of a pair of letters, A with T, C with G, always these pairs, fitting together like gentle puzzle pieces, like hands finding each other in the dark. Why? These 4 letters, why do they matter? Because life is made of limits, and limits make language. Imagine trying to write a poem with just 4 colors, or 4 notes. It sounds impossible, but it is precisely because there are only 4 that the combinations become so rich. Just as music emerges from only 12 notes, and art from 3 primary colors, life emerges from these 4. Adenine, thymine, cytosine, guanine. You don't need to remember the names. Just feel the rhythm. ATC, GAT, CG. The lullaby of life. There is a pattern to the way your body listens to this lullaby. Not all parts of your DNA are read the same way. Some sections are loud, active, genes, others are quiet, silenced, waiting. Some are mysterious, called junk by scientists once, though now we know better. Even the silent spaces hold meaning. In sleep, your body continues to read and repair. Each night is a gentle editing process, as cells check their pages, fix errors, rest, and rebuild. Even now, as your breath slows, your body is working behind the scenes, copying, proofreading, preserving. You don't need to control it. You only need to listen. This is the wonder of being alive, that something so small, so hidden, is guiding you. That the stories of cells and molecules are part of your story too. And while your DNA feels personal, you share almost all of it with others, with other people, with animals, with trees. You and a banana share over 60% of your genes. You and a chimpanzee more than 98%. Even the tiniest bacteria shares some ancient lines of code with you. In this way, you are not separate from the world. You are a chapter in the same long book. Life writes itself over and over, slightly different each time. But the ink remains the same. Tonight you floated gently down into that ink. You traced the shape of your code, the spiral that makes you, and now, just as softly, you begin to drift outward again. You don't need to remember every term, every letter, every loop. All you need to carry is this. You are made of story, a story older than language, deeper than memory, written not in words, but in chemistry. And as you sleep, that story continues, not with fanfare, but with quiet grace. Somewhere, in a distant part of the world, stardust once gave rise to molecules, and those molecules began to arrange themselves. They learned to speak in the language of genes. That is where the next chapter begins. You are drifting still, soft and slow, through the spiral staircase of your own DNA. But now, the steps beneath your feet begin to dissolve, and you're no longer inside just yourself. You are falling backward, gently, weightlessly through time, not days, not decades, but eons. The cells you just visited fade into something smaller, simpler. The spiral unwinds into threads of stardust and atoms, and you arrive at the beginning not of you, but of everything. It starts with stars, in the ancient dark. Before there were cells, or oceans, or memory, there were only elements, hydrogen and helium. The stars, those quiet giants of the sky, fused them into heavier things: carbon, oxygen, nitrogen, phosphorus, the very pieces that would one day compose your blood, your breath, your brain. When the stars grew old, they collapsed. In their final breath, they scattered these new elements across the universe. Stardust, born from death, drifting into nothing, but not for long. That stardust gathered again into planets, into water, into warmth. On one tiny blue world, some of those molecules began to fold into patterns. Some learned to copy themselves. This was the origin of life, not a sudden spark, but a long, slow hush, like a symphony tuning its instruments in the dark. One of the earliest melodies was the language of RNA, a cousin to DNA, single-stranded, simpler, more ancient. These molecules didn't speak with sounds. They folded, they paired, they shaped themselves into tiny tools. Eventually, life found stability in DNA, longer, stronger, easier to preserve. But the echoes of RNA still remain, like an ancestral song still humming beneath the surface. Now the music deepens. Life grew, it changed, it competed, and in time, it learned to protect itself. Bacteria were some of the first life forms to master this survival. Tiny, ancient, everywhere. They may seem simple, but they've been fighting a hidden war for billions of years, not against animals, not against each other, but against something even smaller. Viruses. Yes, viruses are older than you, older than trees, older than bones. They are not truly alive. They are messages wrapped in armor. They cannot grow or eat or move, but they can invade, and when they do, they inject their genetic code into a cell, forcing it to copy the virus instead of itself. A kind of molecular hijacking. A story overwritten that the bacteria needed a defense, something clever, something permanent, and so, over millions of years, they developed a memory. CRISPR, clustered regularly interspaced, short palindromic repeats. A name that sounds sharp and scientific, but think of it more simply. Think of it as a library, a shelf of mugshots, a diary of every viral enemy the bacterium has ever faced. When a virus attacks and fails, the bacterium saves a little piece of that virus's code. It inserts it into its own DNA, like pasting a page into a scrapbook. And the next time that virus returns, the bacterium can recognize it. It uses that saved fragment like a wanted poster, a warning, a weapon. It makes a matching guiding RNA strand that finds the virus and tells a protein exactly where to cut. This protein is called Cas short for CRISPR-associated. One Cas protein is especially famous, Cas9. You'll meet it more closely soon, but for now, just imagine this: a bacterium smaller than a dust mote, with a memory stronger than steel. A living cell, keeping a journal of its battles, learning from each encounter, and protecting its future by editing its own DNA. This is the beginning of CRISPR. Not a machine, not an invention, but a natural immune system, a molecular memoir, and, though you are not a bacterium, the story of CRISPR is now part of your story. 2. Let's drift forward again, not all the way to the present, just a little. Picture Earth, still wild and quiet. Microbes bloom in puddles and vents. The air shifts, evolution hums, and over time, some organisms begin to specialize. They grow eyes, fins, wings, memories, but bacteria remain unchanged in shape, but filled with ancient wisdom. They pass down CRISPR to their descendants like sacred scrolls. It takes billions of years before humans appear. But when they do, they too begin to collect memories, to write stories, to search for the meanings hidden in nature. In the 1980s, scientists studying bacteria began to notice something strange in the bacterial DNA. A pattern, a repetition, little sections of genetic code repeating again and again, separated by unique sequences. They didn't understand it at first. What were these repeating regions? Why were they arranged so carefully? They called them CRISPR, a description of what they saw, but they didn't yet know what they were for. Over time, more clues emerged. Scientists discovered that the strange sequences matched pieces of viral DNA. They began to realize this was not random. This was a defense system. In 2007, researchers finally confirmed it. Bacteria were storing pieces of viruses and using them to recognize and destroy future invaders. It was as if they were building a database of enemies, an internal fingerprint file, a biological memory drive, and soon the most startling idea of all appeared. What if we could use this system just in bacteria but as a tool? What if we could program this molecular guide to cut DNA wherever we chose? This was the turning point. This was when CRISPR stopped being a curiosity and became a revolution. But to reach that point, we must pause. We must return to the feeling beneath this science. You are lying still, listening not just to facts, but to rhythms. And within these rhythms, you begin to sense a pattern. The smallest things often carry the greatest power. A bacterium's memory becomes a doctor's scalpel. A stardust molecule becomes a heartbeat. And the language of life written so quietly becomes a poem. We are only beginning to read aloud. You may be wondering, how can something like CRISPR be precise? How can a cell something invisible, trembling, without thought choose exactly where to cut? The answer lies in pairing, just as it always pairs with T and C always with G. RNA can be crafted to match a specific stretch of DNA. It's like giving the cell a map, a highlighted address, a glowing string that says, here, cut here. The Cas protein, guided by this RNA, moves carefully, like a surgeon with a single task, like a key slipping into a lock. That is the poetry of CRISPR, not chaos, but choreography, and though it began as defense, it is now becoming something more. A tool for editing genes, a way to rewrite the code of life. But tonight, you don't need to worry about the future just yet. You are resting in the quiet past, where cells learned to remember, where survival shaped simplicity into elegance. Outside your window, the world may seem silent, but inside you there is music, deep, molecular music, and in that music there is a promise that what once protected bacteria from darkness might one day help us bring more light, because sometimes a defense becomes a doorway, and just beyond that doorway waits a librarian, a tiny keeper of memory nestled in a cell, and she has stories to show you. Let yourself become small, smaller than you've ever been. Shrink beneath atoms, beneath organelles, beneath thought. You are no longer a person or even a cell. You are drifting now through the soft, invisible folds of the microbial world. It is a world without sound, without light, but rich with purpose. Here, in the warmth of ancient water, a single bacterium floats. It is not impressive to look at. Just a curved rod, so small that millions could dance together on the tip of a pin. But within this quiet body something profound is happening. This bacterium is a librarian. It has no eyes, no hands, no voice, and yet it remembers, not in stories or songs, but in sequences. It carries a tiny shelf of memories rows, of foreign code carefully tucked into its own DNA, that this is its CRISPR array. Imagine opening a book, and each page contains the echo of a threat survived. Not descriptions, not feelings, just the enemies. Name its genetic fingerprint, captured and stored with extraordinary care. Each piece of that stored code is called a spacer, like a Polaroid from a past attack. Between each spacer is a repeat, a little refrain, like the chorus between verses. The repeats make the structure. The spacers hold the memory. This is the bacterial immune system, and it is far older than yours. When a virus invades, it brings its own genetic message, a strand of DNA or RNA that tries to take over the bacterium's machinery, to rewrite the script, to make the bacterium a factory for more viruses. But if this particular invader has been seen before, the librarian inside the bacterium pulls a matching record from its shelves. The matching spacer is transcribed into a delicate copy of that viral mugshot. That guide RNA joins forces with a special protein, Cas9. Together, they form a patrol, a pair of microscopic detectives gliding through the cell. They search for any matching code from the virus. And when they find it, when the guide RNA locks into place, like a puzzle piece, the Cas9 protein cuts a clean, precise slice through the viral DNA the virus cannot replicate. The cell survives, all because a bacterium remembered. This system, simple yet powerful, works without thoughts, without fear, without hesitation. It is life remembering life. You may be wondering, how does the bacterium know which viral fragments to keep? The answer is not perfect. Sometimes it gets it wrong. Sometimes it captures too little or too late. But when it works, that little piece of viral DNA is cut and inserted into the front of the CRISPR array. The newest memory always comes first. Over time, the array grows, a living record of survival, updated like a diary. You float beside it now, this invisible script, this moving archive. It is not elegant, it is not romantic, but it is utterly brilliant. Each CRISPR system is slightly different. Some bacteria have short libraries with only a few entries. Others have arrays stretching to hundreds of spacers, centuries of molecular combat written in silence. These bacteria do not carry shields. They carry stories, and each story helps them survive. But there's another layer to this, another secret. Some viruses evolve to escape recognition. They mutate, changing their code just enough to slip past the CRISPR guides. So the bacteria must adapt to it, becomes a race. A quiet war of memory versus mutation. A biological arms race playing out across oceans, across eons. And in this dance of attack and defense, evolution sharpens itself. Here, in the smallest forms of life, we see the earliest traces of intelligence, not in thought, but in pattern, in memory, in adjustment. And this system is not limited to Cas9. Different bacteria use different Cas proteins. Some are more precise. Some can cut RNA instead of DNA. Some can even edit rather than destroy. These variations are like different tools in a molecular workshop. Some carve, some trim, some rearrange. Scientists are still discovering new versions, like Cas12, Cas13, Cas-X, Casey-H with its own specialty. The librarian, it turns out, has more than one pair of hands, but CRISPR is not only about defense. The more we study it, the more we see its potential for something greater. Because once you understand how the system finds and cuts DNA, you can begin to guide it yourself. You can write your own guide RNA, one that doesn't just defend, but edits, corrects, rewrites. This is where the human story enters. But tonight, you are still here in the microbial quiet, in the mindless wisdom of a bacterium. Let yourself admire it, not as a machine, but as a survivor. It has no emotions, but it carries the genetic echoes of every enemy it has ever faced. That is a kind of grace. You think of libraries as places of silence and thought. Now, imagine a library where each book is a battle won, where each page is a piece of an enemy, pinned gently between lines of repeating code. This is the library that CRISPR builds, and unlike human libraries, this one is alive. It copies itself. It passes its volumes down through generations. It updates itself constantly. Every bacterium born from another carries this library, and though its contents may shift, the structure remains. A rhythm, a repetition, a remembrance. And now you begin to understand how such a small thing could become a revolution. For billions of years, CRISPR served one purpose: survival. But now, humans have borrowed the librarian's keys. They've taken the quiet mechanism of defense and turned it into something else. A scalpel, a pencil, a possibility. CRISPR has stepped out of the bacteria and into the hands of scientists. And in those hands, it becomes more than memory. It becomes design. But design needs tools, and at the heart of this new toolbox are the molecular scissors that shimmer in the dark. Tonight, you enter a different kind of workshop, one without hammers, nails, or sawdust. There is no clang of metal, no smell of wood, just light-soft, pulsing light and an almost perfect silence. Here, the walls are not walls at all, but boundaries made of membranes, delicate, alive. You are drifting once more through the body, not quite in one place, not quite in another. A cell opens before you, warm and translucent. It does not notice you, but it welcomes you in. Inside, something is happening. It begins with a whisperer thread of RNA, gliding like a ribbon through the cytoplasm. This is no ordinary thread. It has been carefully crafted, strand by strand, to carry a very specific pattern, a message, a map, and where there is a map, there is always a traveler. Meet Cas9, not a person, not a machine, but a protein-complex cluster of atoms shaped into something astonishing. Cas9 is not sharp in the way a blade is. It does not slice by force. It slices by precision. It does not guess. It searches. Guided by the strand of RNA, Cas9 moves through the nucleus like a ghost through a library, looking for one particular line of code. Not 2 lines, not 5, just one. When it finds it, it stops. And then it cuts. A single, clean cut through both strands of the DNA's double helix. No sparks, no shattering, just a quiet click, like scissors through silk. This is the miracle of CRISPR-Cas9, a molecular scalpel able to find one sentence in a library of books and make a change. Pause here. Let yourself feel the weight of that. Every cell in your body contains about 3 billion letters of DNA. Cas9 is able to find just the 20 letters it's been programmed to find. Imagine searching for a single typo in a stack of books taller than the moon and doing it in seconds. That is Cas. 9. And this precision is what has changed everything. You float closer now, hovering above the action like a dust mote caught in sunlight. You can see it happening, the DNA unwinding slightly, as if to expose the target site, the guide RNA binding perfectly, the scissors aligning dot, snip dot, a break, a deliberate wound. But this is not destruction, this is invitation, because now the cell must decide how to repair the break, and in that repair lies the magic. Cells have two main ways to fix a broken piece of DNA. The first is called non-homologous end joining. It is fast but messy. The cell simply glues the ends back together. Sometimes it adds or deletes a few letters in the process. This can disrupt the gene, turn it off, silence it, break it. The second method is called homology-directed repair. This one is slower, more elegant. It uses a template, a second piece of DNA with the correct sequence to guide the repair. If scientists provide a template with a desired change, the cell may use it. And just like that, the edit becomes permanent. Change the code, change the cell, and if you change enough cells, you change the body. It sounds like magic, but it is not. It is chemistry. Proteins folding in just the right way. Bonds forming and breaking. Letters replaced, instructions rewritten. This is why CRISPR is not just a tool, it's a language, a way to speak to life in its own tongue. And once we learned that language, we began to ask, what else can we say? Let yourself imagine the possibilities, like lights twinkling along the horizon, a gene that causes blindness, turned off, a mutation that brings disease, replaced with something healthy, a crop that struggles in drought, given the strength to thrive, a mosquito that carries malaria, edited so it cannot spread the parasite. Each snip, each change, like a careful word added to a story, not scribbled in the margins, written directly into the page. You drift slowly through the cell as it begins to repair the break. The strands pull together, tremble, fuse. The double helix reforms, as if nothing ever happened, but something has changed, and that change will echo forward with every new cell that comes from this one. With every copy made, this is inheritance. This is transformation. All from a molecule that acts like a pair of scissors, made not from steel, but from light and memory. Still, even the finest tools require caution. A misstep, and Cas9 might cut the wrong sequence. A near match is not good enough. Even one letter off can have consequences. That's why researchers refine it, sharpen it, study it endlessly. New versions of Cas9 are being developed all the times, smaller, faster, more accurate. There's even a version called Dead Cas9 that doesn't cut at all. It simply binds to DNA and blocks its silencing without slicing. Others fuse Cas9, two enzymes that change letters rather than remove them. These are called base editors, more like pencils than scissors, even finer, even gentler. The molecular workshop is expanding, the tools are multiplying, and still the code listens. You return to Cas9 now, watching it drift back into the cytoplasm, its job complete. It has no pride, no awareness. It is not alive in the way we are, but it holds the power to alter what life becomes. Think of how far we've come from stardust to single cells, to proteins that edit genomes. And now, with CRISPR, the editing is in human hands, the hands of researchers, of doctors, of dreamers. Some use it to study, others to heal. Some whisper of changing future generations. And those questions, They wait for you in another chapter. But tonight, you simply drift through the molecular light, through the careful movements of scissors you cannot see. The cell breathes softly, the code quiets, and as you float outward again, you begin to sense the next story waiting. Not a story of cutting, but of healing. Not a break in the page, but the gentle mending of a line once broken. Breathe in. You are floating again, neither rising nor falling, simply drifting, gently, endlessly, inside the deep library of living things. But now, the pages you see are weathered, worn, some are smudged, others torn, and one by one, you begin to notice tiny errors, letters that don't belong, sentences that don't make sense, loops in the pattern where once there was flow. They are not mistakes in spirit. They are mistakes in code, mutations. Not all mutations are dangerous. Many are harmless. Some are even beautiful freckles, curled lashes, a slightly higher note in the laugh of a child. Evolution is built from them. They are the ink splashes that sometimes become art. But some mutations bring harm. A single letter misplaced, and the body forgets how to fold a protein. A sentence lost, and a cell begins to divide without stopping. A word repeated, and a child is born into pain. Sickle cell anemia, cystic fibrosis, Duchenne muscular dystrophy, Huntington's disease, each one a shadow in the script. Each one a chapter where suffering takes root. Tonight, you are not just reading. You are watching something more tender. A repair. You enter a cell once more. It is quiet here. The nucleus breathes softly, coiled strands of DNA resting like threads in a loom. But one thread is broken. You see eat a gene that should produce a crucial enzyme, but doesn't. One of its letters has slipped. That single letter has caused years of pain in the body this cell belongs to. But now something new arrives. CRISPR, not just as scissors now, but as healer. A guide RNA glides in like a lantern in the dark. Behind it floats cars, 9 steady, waiting. And close behind that, a carefully crafted template, a corrected version of the broken sentence. Together, they approach the flawed gene. The RNA binds to the familiar mistake. Cas9 makes its gentle cut. The strands tremble, split open like pages pulled from a binding. And now the template moves forward. It is not forceful. It is not loud. It simply offers. Here, it seems to say, is how it might be instead. And the cell listens. Its repair machinery awakens. It uses the template to mend the break, not just sealing the wound, but rewriting it with care. When the strands come back together, the typo is gone. One letter quietly changed. The difference between disease and relief, between exhaustion and energy, between a future filled with worry and one filled with breath. You float back from the repair site, the light still glowing. Nothing dramatic has happened. There is no explosion, no transformation. But somewhere in the larger body, something begins to shift. A symptom softens. A struggle eases. A child smiles more easily. This is how healing begins. CRISPR doesn't cure everything, not yet. It cannot reach every cell. It cannot undo all damage. But in the places it touches, it brings precision, hope, change. You drift through other cells now, watching other stories unfold. In one, a muscle begins to grow stronger, where once it failed. In another, a child's lungs begin to clear. In yet another, a blood cell learns to carry oxygen again. Each change begins with a letter. Each letter becomes a possibility. But healing is not just about editing, it is about listening. CRISPR has taught scientists not just how to change DNA, but how to understand it, to see where the patterns break, to find the causes, not just the symptoms, to trace the shape of illness back to its origin, and gently, respectfully, revise. And sometimes the revision is not to remove, but to silence. There are genes that misbehave, ones that produce proteins in the wrong place at the wrong time. With CRISPR, scientists can turn them off, not with violence, but with subtlety. They send in Cas9, not to cut, but to bind, to sit gently on the gene like a stone on a switch. The gene cannot be read, the message never forms, and a storm inside the body begins to settle. Other times, the goal is not to silence, but to add dot, to bring in a new gene, altogether a missing page from the book. With CRISPR, a new sequence can be placed into the genome, like a sentence added, by hand, to restore meaning. Imagine giving cells the instructions they never had, to fight cancer, to resist viruses, to produce missing hormones. This is not fiction. It is now in trials. Doctors have used CRISPR to treat inherited blindness. They've targeted genes linked to heart disease. They've begun editing immune cells to better detect and destroy cancer. And though not every trial succeeds, each one teaches. Each one moves us closer to a world where repair is not rare, but routine. Still, not all repairs are welcome. Not all changes are wise. There is a line between healing the body and redesigning it. That line grows blurry when CRISPR enters the germline, the DNA of eggs, sperm, or embryos. Changes here affect not just one person, but every generation that follows. Imagine editing a story not for one reader, but for an entire lineage. The power is vast, but the responsibility is greater. For now, most scientists hold back. They edit cells in the body, not those that will pass into the future. But the tools are here, the scissors are sharp, and the questions grow louder. Should we fix only what is broken? Or should we try to make something better? And who decides what better means? Tonight, these questions do not need answers. They are clouds that will drift into other chapters. For now, rest in this one. Rest in the quiet of a cell repaired, a page turned, a sentence restored, a life perhaps made easier, softer, longer. Let yourself feel what that means, because even as you lie still, somewhere, in some lab or clinic, a child is being treated, a mutation is being mended, a burden is being lifted. It does not take much, just a guide, a protein, a template, a single change in a sequence 3 billion letters long, and suddenly, a new sentence is born. A sentence that reads more clearly, more gently, more like hope. You watch it take form, glowing inside the nucleus, and as it settles into place, you begin to hear another sound, soft, rustling, like leaves in a field. It is the sound of seeds, and it is growing. You step now into a quieter place. The sterile glow of laboratories fades behind you, replaced by the hush of open fields, the hum of wind through stalks of grain, and the low murmur of water rolling past irrigation stones. You are walking through a garden, but this is no ordinary garden. It stretches far and wide, beyond sight, beyond season. In one direction, there are rows of rice swaying in a monsoon breeze. In another, wheat curls under a dusky sky. Corn climbs skyward. Beans spiral up makeshift poles. and in between it, all quiet. Almost invisible tiny edits are blooming. These plants were not born this way. They were sculpted with CRISPR, not painted, not forced, but gently nudged, one letter at a time, one trait at a time, one hope at a time. You pause beside a tomato vine. The fruit is red, round, gleaming, but hidden inside its cells, a small change has taken root. A tweak to an enzyme pathway that stops it from softening too quickly. It ripens on its own time. It bruises less. It travels farther next to it. A stalk of rice leans gently against the breeze. Its leaves are wider, richer, more efficient. Someone, somewhere, edited a gene that controlled yield. And now, more grains grow per stalk. These are not fantasies. These are happening. Now, in greenhouses in the Netherlands, in test plots in Nigeria. In research centers across Japan and Brazil, seeds are being edited with astonishing care, not to create unnatural things, but to restore resilience. Some plants are made drought tolerant, so they can grow even when the rains forget. Others are edited to resist blights and fungi and viral wilt. Some are made to absorb nutrients more efficiently, some to stay fresh, longer, and in each case, the edit is small. A deletion here, a substitution there. No foreign DNA, no synthetic additions, just the plant. With one less weakness, you walk on. A field of potatoes opens before you. They are quiet, earthy, familiar. But inside, one variety has been edited to resist bruising, another to produce less acrylamide when fried. Farther down, cassava plants grow tall and strong. CRISPR was used to reduce the dangerous cyanide levels that naturally exist in their roots. Now they are safer. And then there are bananas. In East Africa, a bacterial disease once devastated banana farms, threatening food security for millions. But scientists stepped in. They used CRISPR to make a simple change, one that strengthened the plant's immunity. And just like that, a harvest was saved. You begin to understand the gravity of it. Each of these edits doesn't just help a crop, it helps a life. A farmer, a family, a village, a future, because food is not just nourishment, it is culture, identity, memory, security, and when crops fail, people suffer. But here, in this new kind of garden, failure is becoming less common, because someone chose to edit, not to control, but to care. You pause in the shade of a soybean field. Above you, dragonflies hover. Beneath you, roots whisper through soil enriched by small microbial helpers. CRISPR is not only used to edit the crops. It's being used to understand and adjust the bacteria in the soil. 2.2 Fine-tune the entire ecosystem. One scientist edited a strain of rhizobium, a bacterium that helps plants absorb nitrogen. The change made it more efficient, which means fewer fertilizers, which means healthier soil, which means less runoff into rivers, tiny edits, cascading into entire landscapes. You step into a greenhouse next, warm, glassy, humid with breath. Inside, rows of peppers grow in color, red, orange, yellow. Each one carries a small, invisible changer resistance to powdery mildew, a deeper vitamin content, a crispness that lingers. Here, food becomes medicine. Scientists are editing crops, not just to survive, but to nourish more deeply. Rice with more iron, tomatoes with higher antioxidants, bananas with vitamin A. Edits made, not for appearance, but for health. For places where malnutrition still steals energy and vision and growth, CRISPR becomes a quiet ally, not a miracle, not a savior, but a tool that says, what if we could grow food that heals as well as feeds? Outside, the sun lowers over the fields. Birds return to trees. Crickets begin their song. And you feel something you haven't quite felt. Yet on this journey, peace, because in this chapter of the story, the edits are not about fixing broken bodies. They're about nourishing them, preventing suffering before it begins. You walk between rows of maize. The breeze is warm now, brushing your arms. You pass farmers' men and women who walk slowly, checking leaves, humming softly. They may not call it CRISPR, but they see the difference. Their plants survive longer, yield more, require fewer chemicals, grow in harsher places, and they feed their children without fear. That is the quiet revolution. You begin to notice something else. These edits do not shout. They do not glow or sparkle. They do not say, look at I've been engineered. They simply work better. A gene that once responded to heat too strongly is now calmer. A fruit that once cracked open too quickly now stays whole. A leaf that once withered in salt now remains green dot. It's not reinvention, it's refinement. And in many cases, it's no different than what could have happened through natural mutation, except faster, more precisely, without generations of guesswork. Of course, not everyone agrees with this path. Some are cautious. Rightly so. There are questions of control, ownership, ethics. What happens if big companies patent edited seeds? What happens if farmers become dependent on those companies? And so, in this garden, alongside the edits, there must always be trust, transparency, collaboration, respect for culture, for biodiversity. CRISPR gives us power. But how we use IT, that is a choice we make every season. The wind shifts. The sky begins to deepen into violet. And you feel yourself moving again slowly, like a leaf carried downstream past orchards, past research plots, past children harvesting spinach with bare hands and soft laughter. A tomato, plucked and bitten into, juicy and warm from the sun. A sack of rice, full and heavy. A promise held in a seed. You let the garden fade behind you now. You carry its scent, its stillness, its quiet power, and as you drift onward, something begins to return. The questions, what if we could edit not just crops, but ourselves, not just diseases, but destinies? Who decides what should change? Who draws the line between cure and control? In the distance, beyond this garden, you begin to see a fogger place where ethics swirl like mist and choices ripple into generations. That's where you're headed next. But for now, breathe in the scent of possibility and let the seeds sleep. It is quiet now, not the quiet of rest, but of pause, of thought. You've wandered through the spiral staircases of DNA, past molecular scissors and fields of edited grain. But tonight, the air has changed. It carries something else, wonder, and a question that drifts just out of reach. Should we? It is not enough to ask, can we edit genes? We already can. But standing on the edge of what's possible, you must now step into a space that isn't measured by molecules or codes but by values. This is the chapter of choice. Imagine a child not yet born, a small bundle of cells, dividing in the safety of a womb. Within that tiny cluster is an entire future body, a mind, a life not yet lived, and inside those cells, A story is already being written. But what if someone decided to edit that story before it even began? What if we could prevent a devastating disease not after birth, but before? A single letter changed, and a child never suffers. Isn't that mercy? Isn't that love? Yet, what if we change more than that? What if we edit height, eye color, intelligence, muscle strength, mood? Where is the line between healing and enhancing? You float slowly through this fog. It is soft, but dense. And inside it, voices echo. Not loud, not angry, but layered. Scientists, parents, patients, philosophers, they are all asking the same thing. What is a good edit? You come to rest beside a memory. Beijing, 2018. A scientist announces that he has used CRISPR to edit the embryos of twin girls. He removed a gene called CCR5, which HIV uses to enter cells. His goal, he says, was to make them resistant to the virus. But the world does not celebrate. Many recoil. Not because they dislike science, but because of the rush, the secrecy, the breach of a boundary the world had only begun to discuss. The experiment wasn't illegal in every country, but it wasn't ready. Not by a long shot. The edits may have unintended consequences. The gene he targeted affects brain development, not just immunity. And the girls Lulu and Nana will live with edits they did not choose. Their children may inherit them, their children's children too. This is the germline. When you edit a skin cell, the change stays in that cell. But when you edit an embryo, the change becomes permanent. Not just for one person, but for a lineage. And so the question becomes, who decides the traits of people not yet born? You pass now through a room filled with books. These are not molecular this time. They are philosophical. Mary Shelley's Frankenstein, Aldous Huxley's Brave New World, Gattaca, bioethics papers, ancient myths of humans who tried to play God, not because science is evil, but because science, when unanchored from ethics, forgets the soul. Even now, nations are wrestling with regulation. Some allow gene editing for therapy, but not for enhancement. Some ban it altogether. Some wait in silence, unsure, and somewhere in the shadows, private labs whisper of designer babies, catalogues of traits, quiet offers of control. You move through a dreamlike corridor. Along the walls are imagined profiles. Child A, high IQ, fast metabolism, long lifespan. Child B, resistance to depression, high musical aptitude. Child C, low risk of addiction, athletic frame, blue eyes. Each profile a potential life, each edit a preference. You don't judge. You only drift, because it is not your place to decide for others. But it is your place to ask, will we divide society into the edited and the unedited? Will privilege shape biology the way it shapes everything else? Will we come to see some bodies as standard and others as lesser? You pass now through a schoolroom. Children laugh, draw. Some wear glasses, some need wheelchairs. One boy stims with his hands, rocking gently. A girl hums without speaking. Their genomes are unedited, but their presence is whole. And you wonder, will we erase difference in our pursuit of ease? Or will we learn that diversity in DNA is part of what makes humanity vast? CRISPR is not inherently dangerous. It does not seek power. It is a tool, one that listens to the hand that holds it. But like any tool, it can be shaped by the pressures of culture, of fear, of economy. Already, some parents screen embryos for disease. With CRISPR, they may soon edit them instead. Will the line between preventing suffering and chasing perfection begin to blur? And if we remove all that is painful, will we also remove what teaches us to be kind? You drift into a different space. Now a genetics conference, slides, charts, data. A researcher explains that most traits like intelligence or empathy aren't controlled by a single gene. They emerge from networks of genes, influenced by environment, shaped by experience. There is no gene for genius, no edit for happiness. Life is messier than code. CRISPR is powerful. But it is not magic, and yet some still chase the illusion. You walk past a screen showing a list of gene targets: height, metabolism, aggression. Someone has ranked them by editability. It is tempting, so tempting, but temptation is not the same as truth, and truth tells us this: every person is more than their genome. Every life is more than the sum of its edits. CRISPR may shape the body, but it cannot shape the soul. You float now through a garden again. This one is different from before. Here, each plant is slightly imperfect. A vine curves strangely. A leaf has spots. A fruit is uneven. But the air is sweeter, the soil richer, the diversity stronger. A scientist kneels, planting a seed, not an edited one, just a seed. And she smiles, because editing should never replace understanding. It should walk beside it. You begin to feel something stir, a way forward, a path not of fear, nor of unthinking pursuit, but of careful, grounded choice. Ethics, after all, are not chains. They are compasses. They point us not inward, to power, but outward, to meaning. You pass now through a hall of mirrors. Each one shows a version of you, edited to be stronger, edited to be more confident, edited to never forget. But in the last mirror, you see you, as you are, as you've always been. And that reflection smiles back, because perfection is not the goal. Wholeness is. The mist begins to clear now. Ahead lies something brighter, a hospital room, a glowing screen, a patient waiting, a disease with a name, and scientists holding not dreams, but purpose, because CRISPR's greatest promise may not be in designing life, but in preserving it, not altering what makes us human, but giving us more time to live it fully. You enter now not a lab, not a library, not a field, but something quieter, something more human, a hospital. White walls hum with fluorescent stillness. Machines beep in measured rhythm. Voices are soft, shoes muted. You move past waiting rooms, filled with folded hands and half-read magazines, through corridors where charts whisper of diagnosis, and the air carries both urgency and hope. This is where science meets the body, where theory touches skin, where possibility becomes care. And in this still place, CRISPR is learning a new language, the language of healing. You glide into a room where a girl sits beside her mother. Her legs swing slightly. In her hand, a worn stuffed rabbit. She has sickle cell disease, condition written into her DNA like a misstep in a dance. A single change, one letter in the gene for hemoglobin. That's all. And yet, it bends her red blood cells into sickles. Sharp, stiff, slow, they block blood flow, starve her organs, steal her energy. Her laughter dims, not from joy, but from pain. But now, she's here for something different. A trial. Doctors have extracted her stem cells, the source of her blood. Using CRISPR, they've edited a gene to reactivate fetal hemoglobin, a version her body used before birth, one that doesn't sickle. The edited cells will be returned to her, and if they take hold, her blood will begin to flow freely. For the first time in her life, not a mask for symptoms, a rewrite of cause. This is tomorrow's medicine. You drift past another room, where a young man lies quietly beneath a sheet. A disease called Leber's congenital amaurosis has taken his vision, but doctors have injected CRISPR directly into his eye, tiny molecules that snip out a faulty gene and allow his cells to begin reading the instructions they forgot. They don't promise perfection, but already shadows have returned. He sees light, shapes his mother's face. Imagine that. The first thing you see after years of darkness is love. And down another corridor, a woman with cancer sits calmly as her own T-cells, white blood warriors, are infused back into her, edited by CRISPR to better recognize and destroy the tumor that once escaped them. These are not fantasies. These are happening now. Gene editing no longer sits in petri dishes and lab notebooks. It walks the halls. It signs consent forms. It waits. In chairs, doctors once treated symptoms. Now they begin to edit instructions. This does not mean the end of disease, but it may be the beginning of a new kind of medicine. Molecular, personal, precise. You float into a laboratory just above the clinic. The air smells faintly of isopropyl and purpose. Scientists lean over microscopes, not rushed, not distracted. Present. One is editing immune cells to respond to HIV. Another is exploring rare blood disorders in infants. A third is mapping safe ways to edit the lungs of those with cystic fibrosis. The language here is quiet, not dramatic, but filled with wonder. They speak in phrases like ex vivo delivery and adenar-associated vectors. But beneath it, all the real language is care. A gene here, a base pair there. Hope. Everywhere you notice a whiteboard, written across it. First, do no harm. Then, do more good than you imagined possible. It is not always simple. The body is complex. Even a perfect edit can behave differently in one person than another. Some therapies use viral vectors to deliver the editing machinery modified. Viruses that no longer cause harm but carry instructions. Other trials use nanocarriers designed to slip into cells without a trace. Each method must be tested, measured, watched. You peer into a small incubator where patient cells grow in culture. Edited cells, watched for changes, for safety, for surprise. Because medicine is not just what we can do, it is what we should do, and when and how. CRISPR treatments are still expensive, still experimental, still unavailable to many. But the dream is to make them ordinary, to walk into a clinic and say, "My child inherited this, and here we can help." To replace fear with understanding. To swap long hospital stays with short visits. To treat not just illness, but its inheritance. You drift downward now into a digital room. Rows of screens. Bioinformatics at work. Here, CRISPR becomes not just a tool, but a symphony. Algorithms help predict off-target effects. Databases map thousands of mutations. Artificial intelligence scans genomes to find the most effective edits. With the least risk, precision rises. Every trial teaches something new. Every patient becomes a paragraph in a greater medical story. And through it all, consent matters. Equity matters. Access matters. Because if tomorrow's medicine is not shared, it is not medicine. You glide past a chalkboard filled with names, not gene names, human names. Children enrolled in a new clinical trial. Their parents, their stories, each one a life, waiting. There is a sacredness to this science, not because it is divine, but because it is given to researchers by years of study, given to doctors by the trust of patients, given to patients by the hope that suffering might be softened. CRISPR cannot heal every wound. Not yet, not soon. But for some, it is already rewriting tomorrow. You pass a hallway where laughter echoes. A child who once could not run now races through the corridor. Her shoes slap the floor like a drumbeat of joy. Her cells no longer carry the error that once held her still. Her edit has set her free. You turn slowly. Return now toward the quiet of night. Past the glowing monitors, the vials, the refrigerated trays. You pass a nurse leaning gently over a sleeping boy, his fore humming with something new. His genes may be changing even now, but he is dreaming only of stars and you. You are ready to leave the clinic, but something tugs at you just before you go. A question: if we can edit disease, what else might we change? What of the animals around us? The ecosystems, the wild? You feel it again, that shift, that turn, that invitation. Beyond this sterile world, something older calls. The wind through fur, the rhythm of wings, the hum of coral under waves, because CRISPR is reaching them too. And the next story is not just about people, it is about life itself. The world breathes in silence while you listen, not just to one heart or one body, but to an orchestra far grander. You are floating again, this time above oceans and mountains, past tundras and savannas, over canopies so dense they look like thoughts layered in green dot. This is not the story of humans. Not anymore. This is the story of life. CRISPR began in bacteria. It moved through clinics and laboratories. But now it reaches farther. It has stepped into fields and forests, into rivers and reefs, into wings and roots and scales. It is becoming the soft hand at the edge of evolution. You drift toward a coral reef first, beneath the waves. In the blue hush, corals pulse like breathing cities. They are not just stone, they are living animals, hosting microscopic algae that feed them. Together, they form the backbone of marine biodiversity. But the oceans are warming, and the corals are fading, bleaching, dying. Scientists are using CRISPR to help, by editing the genes of the algae, giving them more tolerance to heat, more resistance to stress. They hope to help corals survive longer, stronger. It is not a cure. It is a lifeline. You drift next to a field buzzing with motion. Honeybees spiral through air that smells of pollen and sunlight. But their colonies have been shrinking. Viruses, pesticides, and parasites weaken them. And without bees, so much. Fowls, apples, almonds, wildflowers, balance. CRISPR is being used here to study bee immunity, to adjust genes that help them resist mites, to understand their vulnerability, not to dominate them, but to protect them. In the distance, a flock of birds shifts like a single breath in flight. Some species are disappearing faster than we can name them. Can we help them too? You pass a small, sharp-eyed sparrow. Its population has plummeted from disease. A team is exploring edits to help the next generation resist infection, not in cages, not as experiments, but as guardians. Then your path bends towards something larger, a savanna at dusk. Elephants move like clouds made of memory. They are hunted. For ivory, they reproduce slowly. Some carry diseases that pass into domestic animals. But CRISPR researchers are working gently, here studying fertility genes, modeling elephant cells in labs, seeking ways to preserve their lineage without captivity, to let the wild remain wild. But stronger, you float through a rainforest next. Trees tower above, roots below. Lianas curl like cursive across branches. Some of these trees are close to vanishing. Deforestation, disease, climate shifts, CRISPR, is being used to help certain trees resist blight. Chestnuts in North America, once nearly extinct, are being edited to fight off the fungus that felled their ancestors. Each seed planted is a verse returning to the song. But this symphony is delicate. CRISPR's reach must be careful. One gene altered in a wild population can spread. One change in a mosquito may ripple across continents, because in the wild, DNA is not just code, it is connection. A wolf's gene affects the deer. A deer's movement affects the grass shapes the soil feeds the trees. The trees hold the rain, and the rain returns to the sky. This is the circle. The whole CRISPR may touch one life, but the echo travels. You find yourself in a lab beside tiny fish zebrafish, glowing slightly. They are used in CRISPR studies because their genes are well mapped, and their embryos transparent. But these are not just model organisms. They are part of the pattern. By understanding them, we understand vertebrates. By understanding vertebrates, we understand ourselves. You pass petri dishes filled with frog cells, fruit flies, mice. Every edit here is a question asked of nature. How do you build a spine? How do you remember pain? How do you repair what is broken? And nature answers slowly, molecule by molecule. You drift past research on gene drives. These are CRISPR-powered edits that spread quickly through a population. Scientists have explored gene drives to reduce malaria by editing mosquitoes so they can't carry the parasite. In theory, such a drive could wipe out malaria. In practice, it is still debated, because what happens when a species vanishes? What does the ecosystem lose? What do we fail to foresee? And so, the best minds pause. They test in contained environments. They consult ecologists, ethicists, communities, because CRISPR, even in the wild, must remain humble. It is not our baton. It is an invitation to listen before we tune. And when we do listen, we hear that evolution is not static. It has always been change. But now we are joining the chorus, not shouting, but whispering, helping rice grow taller and bees live longer, helping coral glow stronger and trees stand taller, helping life to live, but always with a question on our lips. What does it mean to intervene in nature's song? Are we harmonizing or rewriting? You float above a frozen tundra. In the distance, woolly mammoth genes glow inside the cells of modern elephants. Some dream of extinction, of reviving lost species, of turning CRISPR into a doorway to the past. It is a thrilling idea, but is it fair? To bring something back into a world it no longer knows. To recreate without remembering why it vanished. CRISPR can do much, but should it do everything? You feel the planet turning slowly beneath you now. Night falls across continents. Bioluminescent plankton shimmer in black waves. Fireflies blink above CRISPR-edited crops. A forest owl watches a rabbit dash. A tree stretches just a little higher into air, now gentler with carbon. Every living thing is a thread in a web that trembles with change, and CRISPR is beginning to pluck at the strands, not to break them, not to bend them, but perhaps to tune them. And still, even as the symphony plays on, you feel the next movement rising, a quieter one, a more intimate one, because, after all the edits, after all the fields and wings and reefs, there remains one question. Who are you in this story? What does your DNA say? And how does it shape the meaning of being human at all? Return now, slowly, gently. Leave the forests, the oceans, the labs. Let go of coral and chromosomes, fields and microscopes. The world hushes around you, like the last page of a book closing softly in your lap. You are back where you began, inside yourself. The story tonight has carried you far from stardust and bacteria to the pulsing heart of medicine, and into the quiet symmetry of the wild. You've seen the molecular scissors, the edited seeds, the ethical clouds, the symphony of life tuned by science. But now, you listen to the most familiar sound, your own breath, in, out, and with each breath, a question lingers in the space around it. What does it mean to be made of DNA, but to be something more than your code? Your genome is vast, 3 billion letters, each strand of DNA unfurling into instructions for how your cells should grow, divide, repair. But you are not just a set of instructions. You are not a spreadsheet. You are not a project. You are a person. You carry memory. You hold pain and joy together like hands inside your chest. You laugh for no reason. You dream while you sleep. You love beyond logic. None of this is written. In your DNA. There is no gene that defines the sound of your voice when you say someone's name. No edit that can mimic the way you forgave someone who never said sorry. No nucleotide that explains the way your chest tightens when you hear a certain song. Your genome may set the tempo, but you choose the rhythm. CRISPR can change your body. It may one day fix disease before it begins, but it cannot teach you what to do with health. It may lengthen your life, but it cannot tell you how to fill your days. Science offers tools, but only you can offer meaning. You are not the passenger in your body, you are the author of your life, and still you carry with you the lives before you, the code you inherited, the mutations and markers, the echoes of people you'll never meet, tribes and tongues and families you will only know through the curl of your hair or the shape of your hands. You are not just the end of a lineage, you are its continuation, and whether or not your genome is ever edited, whether you remain as nature wrote you, or whether a gentle change is made, you remain you. Genes are scaffolding. Your soul is the sculpture. You stretch now inside this thought, this body that is both ancient and new. Your cells are reading DNA right now, making proteins, sustaining life. And yet you are still listening, not moving, just being. This is where CRISPR ends, and where you begin, because for all its precision, all its promise, CRISPR is still a tool. It can cut, it can repair, it can design, but it cannot choose. You do. You choose kindness. You choose courage. You choose to ask questions. You choose to pause, like now, and wonder. CRISPR may let us touch the roots of life, but only you can touch its meaning, And what is meaning, if not the sum of our small choices, repeated quietly across time? The doctor who sits a little longer with a frightened patient, the farmer who saves a handful of native seeds, the scientist who pauses before editing something that cannot be undone. These are not code. They are character. You drift through your own cells, now billions of them, each one humming, each one carrying the same spiral of DNA, yet doing something different. Some build your muscles, some feed your brain, some defend your blood, some line your lungs, same instructions, different interpretations, like musicians reading the same sheet music, but adding their own emphasis, their own silence between the notes. That's what makes the body alive. Not just the code, but the nuance. And perhaps that is true of people. To dot we are given our beginnings, but we shape our endings. And in the space between the middle, we create something no tool, no editor, no algorithm can replicate. A life. You stretch into that middle now, not with urgency, not with answers, but with peace. Let this chapter close, not with closure, but with expansion. Let yourself widen into the mystery. CRISPR has shown you what can be changed, but tonight you rest in what must remain. The breath, the heartbeat, the silence, the song. You are the story that will never be fully mapped, a question that cannot be resolved, and still a part of a greater whole, just like every cell in your body, every voice in the world, every letter in the code. You carry the memory of stardust, the promise of future edits, the weight of generations, the lightness of this moment. And as sleep begins to fold around you like soft paper, you hear, faintly, the CRISPR spiral still, turning not to define you, but to remind you that your life is the only genome ever written like this.